So thank you very much for having me. It's really nice to come to this. I think, as Simon kind of underscored, I feel it's really important that we recognize ourselves as a community, perhaps a distributed one, but a community nonetheless. And it's important that we communicate with one another and share information with one another. So I think if Elon Musk had his way and deposited us on some faraway planet, one of the first things humans would do would be to explore. They would try to figure out exactly what's on that planet. And in our own case, here on planet Earth, there's a couple 100 years, 300 years maybe, of explorers out there trying to figure out the multicellular life forms on this planet. But when it comes to microbial life forms, really it's DNA sequencing and the efforts of people in this room and elsewhere over the last couple of decades that have really started this initiative, which I'm going to call the Great Experiment. And what we found is when we've gone out there and looked for microbes and viruses, we found that we're literally floating in a world of viruses. Moreover, they live all inside of us. So they're critical, as Eugene was stating earlier, to probably everything in terms of evolution. They're critical, as Matt was stating earlier, to probably everything that concerns how biosystems work. And so the question is, how do we go from here? And we're just early days now. And when you think about the big experiment, we're talking about the things that Eugene's talking about, while at the same time, the things that others were talking about today, that is the long-range evolutionary impact, as well as the short-range evolutionary impact. So the folks that study influenza are interested in how influenza evolves from year to year, whereas the folks who are studying polytons are interested in how DNA elements have evolved over millions of billions of years. So how do we do this? Well, NCBI, for better or worse, is sort of at the center of the data universe, literally by design. And so they're uniquely suited to be an aggregator and distributor of publicly available data. So anything else is really kind of gravy. And the question is, can we use this unique situation to really leverage this data and support this big experiment? Because this big experiment is going to take decades. And is there some place where the data can go and can be housed and can be kept up to date and can be put into sets that are useful to people so they can get something out of the data? And that's really the sort of the idea behind what we're calling the NCBI Virus Project, and that is to look holistically at the data universe, try to bring data into NCBI from all sorts of places, and then individual submitters to big institutions, and then try to keep that data up to date and organize that data and then push it back to people so it's useful. And so this model is a little bit different than you've heard probably people from NCBI talk about before, because the model really includes everything from submission to retrieval. And so in the past, when you hear about submission, you think about GENBANK. When you hear about processing, you might hear about some data validation projects like RefSeq. And when you talk about retrieval, you hear about things like Entree and FTP. And so what I'm going to talk about this talk is how we're trying to get rid of that idea that there's these different places. And we're trying to get to an idea where it's all one thing. It's a model that works together holistically. So this concept is not particularly new. As people started seeing GenBank flooding with sequence records over the past decade or so, they realized it was going to be really important to process this information, figure out what sequences were good sequences, what sequences were bad sequences, and make those sequences available to people. And that kind of gave rise to the NCBI Viral Genomes Project, which is where most of you guys probably met me. And there we were trying to basically pick out some reference sequences that sort of explain the taxonomic universe as it was known. We tried to go and find genomes that we thought met some set of standards. We tried to improve the taxonomy of those genomes. And we tried to index those genomes in a number of different ways, including among host groups and things like that. And basically, we ended up with this project spending a lot of time on taxonomy, because that turned out to be the one key thing that everybody needed to know, but that was really poorly described at the time. So I've spent about 9 years since I've been at NCBI doing a lot of taxonomy, which is really funny because I'm a biochemist that used to work on T4, so taxonomy is something very foreign to me. But I did, and I love Eugene's work, and because, there's this idea of what humans want out of taxonomy. And on the one hand, we want to know about our distant ancestors in a way, and in another way, we want to know what's in my tube today. And trying to find sort of a commonality in those two use cases has been very interesting. And that's gotten us into the viral taxonomy business. And you'll see here that we've got about twice as many NCBI viral species as there are ICTV viral species. And that's about 50% of all the claimed viral species that have been submitted to GenBank. So we've been working very hard with ICTV to do this, to improve taxonomy. We also realized early on that people really care about the host from which viruses came from. So we started this manual process of putting viral sequences into these host type boxes. And what came out of that was, if you're really good at Entree, you can go to one of our databases and you can use these sort of index terms up here. And I'm sure everyone here knows those, right? There are 5 people here, I do know them because I send these to them, right? And then if you do this, you can find exactly what you want, right? And so that's kind of a fail. And so we built this homepage, which was an attempt to aggregate data. And each one of these links turns out being some sort of separate resource to hold data in a certain way for a certain group. And then we have this site down here, which is basically just an accession list of all our validated viral genomes at NCBI, about 120,000 viral genomes, and some metadata associated with them for the heavy users. And we sort of realized this was a fail, and it's just not good enough. And so the question is, what do we need to do to make it better? And really was the focus on the processing side of things to normalize the data better, and then also on the retrieval side to make it easy to find the data. And this was really the sort of the founding sort of ideas behind the Virus Variation Project, where instead of employing a lot of human tools, we employed a lot of processing tools, computer tools that would automatically taxonomically bin sequences, would annotate those sequences, would store normalized metadata. So rather, if the record says Homo sapiens, or it says patient, or it says little girl, that all gets mapped to Homo sapiens, and then we have common terms that we pull off that hierarchy. And basically using tools, computer tools, as best we could to do the 95% of the data processing, and then to allow the humans to look at the other 5% of the edge cases. And then finally, we wanted to package this whole thing into a sequence selection interface. And this project's gone through a couple iterations. I'm going to show you the latest. Oh, I'm going to show you this first. We focused on 7 pathogens for obvious reasons because we're National Institutes of Health. And all I want to try to show you with this slide is that when we apply this processing to very well-studied clinical viruses where you think everything should be great, right? Where you think that there should be established standards. The bottom line is we remap a whole bunch of data, right? And so remapping all this data allows us to support this interface here that was done by a group at NCBI, and it's just a prototype. I promise you won't get this many checkboxes if you come to our pages. But I want you to show these checkboxes are all normalized data fields. So you can come in here and, press a single box that says human and get all the human viral sequences. And so this is sort of the direction we're heading, making it simple to find fairly complex data sets. And so you get a table back, and that table shows you everything that's been mapped, all the the attributes for host and the sample itself. And there's actually like 20 fields that you can select in this table. But the bottom line is you can just download from this page. So you pick your format and you download. So your search is easy, your download's easy, and you're done, right? And so now we've gone back to what was our viral genomes. And then so what we have now is between our viral genome set and this virus variation data set, we have, I think, 700, 800,000 GenBank records that have been processed this way. And so that's a fairly large percentage of the 2.6 million GenBank records total. So we're really making taking a large chunk of data and normalizing it. And we're now taking that viral genomes data I talked about before that was all manually curated at some point, and we're running that through our processing. And I think the take home here is we can do it, but we need some work on this interface, because when you have that many checkboxes, it's sort of a fail. And this part is under construction, but the back end is up and running, and we're going to have these things in these interfaces. All our validated data will be available through these interfaces soon. So what does this matter? Well, it matters on that data retrieval side, because I can go and easily search and find my sequences, right, and download them. But it also matters on the data analysis side. So here's a little tool we've now built and prototyping right now. Some of you may have been asked to prototype it. Basically, it's BLAST for viruses. It's nothing fancy or new. We've got a BLAST interface. But when you get your hits back, you get them back in this table of all your normalized metadata. So if you've got, a bunch of stuff in the tube you built some assemblies from, and you want to figure out something about those assemblies, you go to a tool like this and you find out, wow, this thing's been found in this country and been associated with this host. And you immediately know something, rather than going and trying to click 15 different, return GenBank record hits and normal blasts, right? So I see this normalized data as not just a way to empower people to go search things and find things, but also as a way to empower their own analysis, right? So I've been talking about GenBank and assembled records, but really that's not my favorite thing. My favorite thing is read data. And it's sort of, I was an early adapter to using sequencing as an assay, early 2000s. And I've always been fascinated by the read data and the technology associated with it. And the problem with the read data is just too darn big. And I know people who have crazy access to crazy computers come up to me and tell me there's just too many read sets in SRA. And so we're really working on some ways to improve access to SRA data. And we focused on the retrieval, and this is how, actually talking to people works, because people in this room bugged me about it. And so we said, well, we could do a better job of helping you find these sets. And we started off with some simple things we were doing behind the scenes, and those became a tool. And this is one of those tools. This is what we're calling our SRA lookup tool. It's based on some technology we're calling STAT. It's unimportant. It's a K-mer index. It's similar to Kraken. We're doing some fancy stuff to make it smaller, and I'd be happy to talk to people about that in detail. But for me, the dumb biologist often in the room, all I care about is I can take a sequence and I can search it against SRA, and lo and behold, I can find runs that have that sequence in it, right? Basically what it's doing is it's finding K-mers in your sequence and going and finding run sets that have those K-mer values. And we built a fairly enormous index to support this. Going back to something Eugene said earlier, one of the people that convinced me to do this was Rob Edwards, because he wanted to find instances of crassphage, and he was sure it was out there. And sure enough, we did some stuff behind the scenes, and we're like, Rob, you're right. As you can see, there are tens of thousands of run sets that have the K-mers associated with crassphage in them. So we see this as a great exploratory tool, but more importantly, the tip of the iceberg for other tools that we're going to develop around SRA data. And finally, I just wanted to end on this point. So I'm talking so much now about stuff that's been submitted through traditional methods into NCBI. But that shouldn't be the wall, right? So when we start talking about read data, there's lots of different databases out there that house tons of read data. And some of that data is interesting, and some of it is really important, if not critical, to the community. So what I'm going around this year talking about, I'm trying to bring more people into a group that want to bring more data into NCBI. So if you have hundreds of thousands of sequences, but there's some 2,000, 3,000, they're really important because they may be the first finding of a particularly unique taxonomic class, or they may have some interesting biology associated. We want to bring them into our constructs. So we're looking at ways to submit these things and to house and to store them. And of course, there are lots of people in this room that are building assemblies out of as third-party users of publicly available data. We want to find ways of storing those important assemblies. And a lot of this is going to require community standards and everybody coming together and talking about it and what we really want out of the data. But I think we can do it. I think this is something that can be achieved within the next few years, actually. lowering the burden and coming up with the process that gives the data out at the very end that people really want. So finally, I just want to acknowledge a bunch of people, particularly Emin Bao and Sergei Zhdanov, who were key members of the group up until very recently as they've gone on to do bigger and better things. And the folks, Eric Nowicki and Alejandro Schaefer, working on our annotation tools, and Louis and Lani, who are working on our interfaces with us. Thank you very much.